Okay, great, awesome. Okay, well, first I'd like to thank the organizers and Rod for putting this together and inviting me to participate in this awesome symposium on type 2 immunity and allergic disease. So this is actually a relatively new area for my lab. Unlike Dr. Sokol, this has not been something that I've been thinking about for my scientific career. So it's really fun to be able to participate and share with you how we got into this and a little bit about our recent work. So the question that I personally have been interested in for a long time now is how do cells differentiate, which is really just a basic cell biological question. How does a cell that has more potential ultimately receive signals and become a more specialized cell through the process of differentiation? And what are the signals that drive that? And the immune system is a great model system to look at that kind of a question, partially because, can you guys see my pointer? Yes, okay. So as you can see here, in the immune system, you have hematopoietic stem cells, and these can move into these different lineages that will drive the immune cell, the different immune cells here. But as many of you know who are immunologists, these sort of terminal fates are not really a terminal fate at all. In fact, many of these cells are still circulating through the body and waiting for additional signals so that they can further specialize into different subsets. So in the context of T cells, these cells can go on to become a variety of different subsets which have individual and unique functions. And so these are the kinds of questions that I've been interested in for a long time, or what are the signals that will drive different cell types to differentiate in response to their environment, and what are the outcomes of those functional subsets that are important for that environmental signal that they receive. So more recently, we've been interested in this in the context of tissue-specific immunity. So of course, the tissues, as Carrie just very nicely introduced, are both the site of where pathogens or allergens are introduced, and then they're also the site where you get a lot of that inflammatory responses. And so in the context of T cell function, those pathogens or allergens are exposed to the tissues, and then as Carrie nicely showed, they will be transported by dendritic cells, or they can drain directly into the secondary lymphoid organs. And this is the site where that T cell differentiation is going to be primed and initiated. Once they become effector cells, they will then migrate back to those tissues where they can then go on to mediate that endpoint inflammation. And so this is the context of sort of the environment, the tissue environment in which T cells become activated is something that we're now sort of beginning to think more carefully about. And I'll come back to that a little bit more at the end of my talk. So going back to the concept of T cell differentiation. So this is a slide that I've been using for probably my whole career, thinking about how transcription factors play an important role in controlling T cell differentiation. So you can sort of split the transcription factors up into two categories. The STAT family of transcription factors are constitutively expressed in naive T cells and are triggered rapidly in response to cytokine stimulation. And depending on the STAT that becomes activated, it will trigger activation, upregulation, and expression of master regulators of transcription factors shown here, which are specific transcription factors that are considered necessary and sufficient for the differentiation of these specific functional subsets. So for example, Th1 cells require TBAT, and they will make interferon gamma. And that interferon gamma is a critical part of the Th1 functional outcome. Today, of course, we're talking a lot about type 2 immunity. So GATA3 is the master regulator of Th2 cells, and STAT6 is the major STAT that drives that. And that will go on to drive cytokines such as IL-4, IL-13, and IL-5, which have a critical role in that type 2 inflammatory response that we'll be talking about today. However, there's another category of transcription factors that do play a role in T cells, and those are transcription factors that are not sort of specific. So the transcriptional factor that I've been thinking about for a long time is actually a pleiotropic transcriptional repressor called Blimp-1. So Blimp-1 was initially identified in B cells as the master regulator of plasma cells, but is also expressed in many cell types, even outside the immune system, including trophoblasts, so that's required for placental formation. In the context of T cells, Blimp-1 can be expressed in each of these different subsets with the exception of the T follicular helper cells. So Blimp-1 is a potent repressor of BCL6, which is the master regulator for TFH cells, but can be expressed in a subset of each of these different CD4 T cell subsets. And the expression of Blimp-1 in each of these populations tends to associate with enhanced or additional effector functions, such as the expression of IL-10, or in the context of Th17's GM-CSF and interferon gamma. So my lab has been interested in some time in sort of a basic question of how is Blimp-1 turned on in T cells during T cell differentiation and in their effector functions, and what is Blimp-1 doing specifically in each of these cell types? So we got interested in type 2 cells and understanding what are the drivers of Blimp-1 in Th2 cells and what are they specifically, and what is it specifically doing there? More broadly, the importance of Blimp-1 in T cells was described over 10 years ago when two groups independently made T cell specific Blimp-1 knockout mice. And those mice developed relatively rapidly within a few weeks a multi-organ inflammatory disease that was really pronounced in the colon, leading to a wasting disease, a fatal wasting disease, and autoimmune colitis. And so that's really shown here, where you can see wild type mice have this, very normal colon architecture with the epithelial cells, but in the absence of Blimp-1 in the T cell compartment specifically, it leads to an increased inflammation in the epithelial cells, lymphocyte infiltration, and you can see total disruption of the architecture. And that led to a fatal wasting disease. So these data suggested that Blimp-1 was critical in T cells to constrain T cell mediated systemic autoimmunity, because again, this was a spontaneous response. So based on these data, we asked whether Blimp-1 in T cells could also control responses to non-infectious stimuli such as allergens, because of course, allergens are not pathogenic. And so we postulated that perhaps Blimp-1 might be also important for constraining responses to these non-infectious stimuli. So thinking about a type 2 response, we decided to turn to allergic asthma as a model where we could test the role for Blimp-1 in a type 2 allergic response. So a little bit about allergic asthma. It is a type 2 T cell mediated disease. Th2 cells are known to be a critical component of the response, much as they are in the cutaneous response that Dr. Sokol just discussed. And so in this case, inhaled allergens such as house dust mite or cockroach extract or tree pollen interact with the epithelial cells in your lung, which drives a variety of inflammatory responses that will lead to a Th2 response, which drives IL-13, IL-4, and IL-5, and that leads to recruitment of eosinophils in the tissue, bronchial hyperreactivity, globule cell metaplasia, which leads to mucus production in the airways, as well as IgE, allergen-specific IgE, which is, of course, IgE is a hallmark of type 2 allergic responses. And so Th2 cells in the context of allergic asthma and the development is still known to require STAT6 and IL-4. However, many non-canonical pathways have been shown to support Th2 development in the context of allergens. And as Carrie just discussed, how this response occurs, how is it that allergens that you inhale really promote a type 2 response is still not really well understood. So this is an area of investigation by a variety of labs. So for us, we wanted to test again the role of Blimp-1, and our hypothesis was that it would be involved in constraining responses. And so we thought that in the setting of an allergic response like this, loss of Blimp-1 in the T cells would lead to more effector cells, more effector Th2 cells, and worse disease, much like you saw in the colon where you had a pronounced inflammatory response. So to start looking at allergic asthma, we partnered with Dr. Honorado Ray, who's here at the University of Pittsburgh and is an expert in type 2 allergic responses, particularly in allergic asthma. And so with her help, she helped us get up and running with a chronic inhaled house dust mite model. So in this model, we take either control mice or mice that are specifically lacking Blimp-1 in the T cell compartment by crossing them to CD4 Cre. These are Blimp-1 flocks crossed to CD4 Cre mice. And we sensitize with daily intranasal immunizations of house dust mite. We rest for 4 days, re-challenge for 4 days, rest for 3 days, and then re-challenge again. And then this is the endpoint. And if you just look in the lung tissue at this endpoint in control mice, you can see that the lung tissue has a lot of lymphocytic infiltration. The airways are starting to get sort of inflamed. They have pronounced increases in size. And then this pink staining here, this is called a PAS stain, is an indication of mucus. So you can see there's mucus starting to develop in the airways. So these are key hallmarks in the lung tissue of an allergic response to the house dust mite. Much to our surprise, the Blimp-1-deficient mice had absolutely none of those characteristics. In fact, the lungs looked like they just were completely normal. We had no infiltration and no mucus and no thickening of the airways. When we looked at the bronchoalveolar lavage to see what kind of cells were being recruited into the lung tissue, We saw a specific decrease in the eosinophil compartment, indicative of a loss of type 2 immunity potentially, as well as a decrease in airway hyperresponsiveness. So basically, the loss of Blimp-1 in the T cells seemed to be completely protective in terms of the allergic response, which was a total contrast to our expectation and our hypothesis. So we wanted to dive in and next look at the T cells in the lung tissue to try and get a sense of what might be going on here. And indeed, sort of supporting the specific loss of the eosinophils that were being recruited to the lung tissue, we saw a specific loss of the Th2 cell compartment, whereas the Th1 cells compared to control mice and the Th17 cells compared to control mice were fairly consistent. In contrast to the Th2 cells being specifically decreased in the absence of Blimp-1, we saw an upregulation of Tregs. This was actually not a surprise to us. There have been previous reports that the loss of Blimp-1 in the T cell compartment leads to an increase in Treg cells. This is thought to be due to the fact that Blimp-1 can repress the IL-2 gene. And so in the absence of Blimp, you get more IL-2 in the system. And of course, Tregs love IL-2. So any system where you have excess IL-2, you often will get excess Tregs. Nonetheless, we next wanted to ask whether this loss of Th2 cells was due to intrinsic Blimp-1 loss or whether it was due to the increases in Tregs that were suppressing the Th2 cells. So to answer that question, we generated 50-50 mixed bone marrow chimeras. And then after these were reconstituted, we subjected those 50-50 chimeras to the house dust mite and analyzed the response. And what we found was very clear. All of the bone marrow that generated T cells that were Blimp-1 deficient were unable to form Th2 cells, whereas the control bone marrow could form Th2 cells fine. However, the Tregs were now normalized, suggesting that it was in fact intrinsic Blimp-1 that was leading to a decrease in Th2 cells and an extrinsic effect that led to an increase in Tregs. So this made it clear to us that it was intrinsic Blimp-1 that was important for Th2 cell development and not due to an increase in Tregs that was then suppressing the Th2 cells. So as I mentioned earlier, IgE is a hallmark of the allergic response. So we looked to see if that was affected, and in fact, it was not affected. So the loss of Th2 cells had no impact in the setting for IgE responses. And this was perhaps not really much of a surprise because we now understand that T follicular helper cells are the main T cells that engage with B cells and drive B cell responses. So in the absence of Blimp-1 in the T cells, we again see this very specific loss of Th2 cells. This is now looking in the draining mediastinal lymph node from the lung. And in comparison, the T follicular helper cells, which are IL-4 positive, as well as CXCR5 and BCL6 positive, there was no change in the amount of TFHs, which led to a normal amount of IgE responses. So really what Blimp-1 was doing here was sort of molecularly dissecting the TFH from the Th2 response, which allowed us to really see that it was specifically promoting Th2 cell development. So the next question we wanted to ask was, how is it that Blimp-1 controls Th2 cell differentiation? So we got a little bit of a hint from the fact that while GATA3 was specifically lost in the Th2 cells, BCL6, which is known, Blimp-1 is known to repress, was upregulated. And so interestingly, BCL6 is also a known repressor of GATA3. So it suggested to us that this increase in BCL6 was maybe responsible intrinsically for limiting GATA3 expression. So to test if that would be true, we asked if deleting BCL6 from the Blimp-1 deficient T cells would restore Th2 cells and allergic asthma. So to answer that question, we were fortunate enough to obtain double conditional knockout mice that were T cell specific from our collaborator Alex Stent at Indiana University. So these are BCL6 floxed, Blimp-1 floxed mice crossed to CD4 T, so they're lacking both. And we compared them to our single knockouts. And indeed, as you can see here, while the Blimp-1 single knockouts, again, had very limited infiltration in the lung tissue in the context of house dust mite-induced allergic asthma, the double conditional knockouts, much like the BCL6 single knockouts or the controls, had restored lymphocytic infiltration as well as mucus in the airways and really a robust histology consistent with inflammation. We also were able to restore eosinophil recruitment to the bowel, as well as specifically restore Th2 cells into the lung tissue. So Th1 cells looked very similar to the Blimp-1 knockouts, the single knockouts, and the increase in Tregs was also very similar to the increase in Blimp-1 knockouts. However, Th2 specifically were able to come back. And then of course, this was kind of not also unexpected that the IgE response would actually track with BCL6. So the loss of BCL6, which leads to a loss of T follicular helper cells, is what's really controlling the IgE response here. So this suggested to us that the mechanism by which Blimp-1 was acting was to repress BCL6, which is a repressor of GABA3. Okay, so this was very interesting. We had previously published that IL-10 via STAT3 was an axis in Th2 cells to promote Blimp-1. And in the setting that we looked at in our previous publication, which was mainly in vitro or in a worm antigen model, we found that loss of Blimp-1 actually did lead to an increase in Th2 cells, and that we had some evidence that Blimp could antagonize STAT5 and lead to cell death. But we were really kind of interested, based on this previous data that we had that said IL-10 through STAT3 was a driver of Blimp-1, as to whether or not STAT3 might also be a driver in allergic asthma to promote Blimp-1 in Th2 cells. So to answer that question, we utilized STAT3 conditional knockouts in the same model that I previously described. And indeed, we saw almost the exact phenotype that we saw with our Blimp conditional knockouts: a loss of eosinophils, a loss of Th2 cells, as well as a decrease in Blimp-1 expression in the STAT3 knockouts, suggesting that STAT3 was a direct driver of Blimp-1 in this Th2 system. Okay, so then what is potentially activating STAT3 in our T cells? So in the context of T cells, there's sort of three main cytokines that we think of that can activate STAT3: IL-6, IL-21, and IL-10. So although we had previously shown that IL-10 was important for driving STAT3, we actually focused our efforts first on IL-6 and IL-21, primarily because IL-10 is a well-known anti-inflammatory cytokine. And it was hard for us to conceptualize in this setting how an anti-inflammatory cytokine would be promoting allergic disease. So we started by looking at IL-6 by using IL-6 receptor alpha T cell conditional knockout mice. And unfortunately, we saw no change in Th2 cells and no change in Blimp-1 expression, suggesting it was not IL-6 that was driving Blimp-1. So we next turned to IL-21 receptor deficient mice to look at the role of IL-21. And we were actually initially hopeful because similar to data that Bart Lambert had published in 2015, we did see a complete loss of Th2 cells in the absence of IL-21 receptor signaling and a loss of eosinophils that were recruited to the lung tissue. However, we didn't see any change in Blimp-1 expression in the absence of IL-21. So however IL-21 is promoting Th2 cells, it's independent of the role of Blimp-1 in our setting. So this kind of led us back to IL-10. So we decided, okay, let's see if this is in fact IL-10 as we had previously published. So to do this, we first used Blimp-1 reporter mice, which report Blimp-1 expression using YFP. And we treated the mice with an anti-IL-10 receptor blockade antibody in the context of the house dust mite allergic asthma response. And indeed, we found a specific decrease in the Th2 cells, in the eosinophils, and in Blimp-1 expression in the Th2 cells tracked by YFP expression. So this suggested that in this setting, IL-10 could actually promote Blimp-1 and promote Th2 cells and promote eosinophils in the lung tissue, which was really very surprising to us. Now, of course, IL-10 is also a very pleiotropic cytokine. It can act on a number of cells as well as come from a number of cells. We wanted to confirm this in IL-10 receptor alpha deficient mice to see if IL-10 was in fact directly signaling to the T cells to promote Blimp-1 expression. So we used IL-10 receptor alpha deficient T cell specific knockout mice. And again, we saw a decrease in Th2 cells as well as a decrease in Blimp-1, suggesting that in this case, IL-10 is in fact promoting Blimp-1 and ultimately promoting Th2 cells and allergic asthma response, in contrast to its well-described role as an anti-inflammatory cytokine. So as you can tell, we obviously found these data very surprising. Blimp-1 is not required for Th2 cells in vitro. We've shown that, other people have shown that. That data is shown here. This is just doing general T cell differentiation in vitro skewing. If you look at the Th2 cells in the absence of Blimp-1, there's really no impact. Th1 cells are also largely normal as well. And as I mentioned, we had previously shown using a subcutaneous injection of soluble worm antigen, which is derived from Schistosome mansoni, a worm. Of course, worms are an important driver of the type 2 response as well. We saw what we sort of had initially expected to see with our allergic asthma response, that Blimp-1 could constrain effector responses, so that in the setting where Blimp-1 is deficient, you actually get more Th2 cells in this case. So this kind of led us to this question of what is supporting Blimp-1 driven Th2 development in the context of inhaled allergens? Is this due to the route of immunization, or is this due to the antigen itself? So to try and address that, the first thing we did was go back to our SEA model, but now perform it intranasally in the same way that we did with our house dust mite. And what we found is that, yes, much like the house dust mite induced allergic asthma, SEA given intranasally also led to Blimp-1 was required for Th2 cells. So this was very similar to what we saw with house dust mite, suggesting that really it was potentially the route of immunization, not the antigen, that was promoting this Blimp-1-dependent Th2 pathway. We next sort of confirmed this by taking the house dust mite and doing it subcutaneously. So we either did this in the foot pad or in the flank. Either way, it led to the same result, that in this setting, when you put an allergen subcutaneously in the skin, now Blimp-1 is required to constrain effector responses, as shown here, much like our initial hypothesis. So lastly, we wanted to confirm that this wasn't just a difference between looking in the draining lymph node versus looking in the lung tissue in these two models. So we turned to sort of a more traditional method of generating allergic asthma by doing an OVA plus alum IP prime, 2 days, 7 days apart, followed by a 1% OVA challenge via nebulizer. And in this setting, now we're again looking at Th2 cells in the lung. We really don't see any difference in the percentage of Th2 cells, whether you have Blimp-1 or not. So really what these data are suggesting to us is that when your allergen is priming via the lung tissue, via that intranasal route, that's the setting by which Blimp-1 is required for Th2 cell development. So just to conclude, this is sort of the graphical abstract that we had for this paper, which was published in Exp Med last year. Inhaled allergens, we believe, are driving IL-10 to promote T cells to drive Blimp-1-dependent Th2 responses, which can then migrate back to the lung and drive increased lymphocyte infiltration and eosinophils, as well as airway hyperreactivity. And the mechanism of this is by repressing BCL6, which is a repressor of GATA3, so indirectly promoting GATA3. In the absence of Blimp-1, you lose this Th2 response. You have normal cellular infiltrate and no airway hyperreactivity. However, the T follicular helper cells and the IgE response is intact. So some of the future directions, we're obviously really interested in trying to understand the source and the location of IL-10 to promote T cells. We really are trying to understand whether Blimp-1 is coming on at this point in the lymph node, or whether this is sort of an early undifferentiated state, and then it's upon migration back to the lung tissue where Blimp-1 comes on, and whether the IL-10 is here. We have some early evidence to suggest that in the setting of lung intranasal immunization, IL-10 can be found in the lymph nodes and may be driving Blimp-1 in that way. We're also interested in looking at the role of Blimp-1 in tissue-resident memory cells, and I think that's something Rod's going to talk about next. But Blimp-1 is known to be important for TRM maintenance. And because, of course, this is an allergic response that's chronic, we want to understand whether Blimp-1 can also have a role in driving TRM redifferentiation into Th2 cells in the context of that chronic rechallenge response. And then lastly, we're really interested in sort of what are these lung-specific properties that can support Blimp-1-mediated Th2 cell development, while systemic or skin-mediated responses do not. And so I think with that, I'm actually going to just mention that some of the tissue-specific factors that we're starting to consider are the different dendritic cells and macrophages that might be coming either from the lung or from the skin, as well as maybe the neurons that are present in the lung or the skin, based on the work that Dr. Sokol just presented, as well as work from Dan Kaplan and others that are indicating that these different neurons can play an important role in T-cell immunity. And so I'll just quickly thank my lab. This work was primarily done by a very talented technician, Angela Hettinga, who's already moved on from my lab to start nursing school, as well as a very talented postdoc who joined my lab, Dr. Kunhi, and is doing a lot of follow-up studies on IL-10 as well as TRMs. We couldn't have done this without our wonderful collaborators. Anurada has been instrumental in helping us get into this field and interested in how to do these assays, and the double conditional knockouts from Alex Dent. And I'd be happy to take any questions.
So thank you very much, Dr. Amanda, for this great talk. So if anyone in the audience have any questions, so please type it in the chat. I think I will go with the first one. I was looking at some data also in the Blimp animal that there are some data that indicates that in lung inflammation, not having Blimp will increase you Th9 cells. In fact, you will generate exacerbation of inflammation. And now again, we come back to the point that in that particular setup, the antigens was given by intraperitoneal, and then So I was thinking also if in your particular condition or experimental setup, Th9 cells will be also affected.
So it's a good question. We have not looked at Th9 cells. We just haven't done it. It's sort of on our list of things to do. But yes, I think that the key point between that study and our study is that in that study, they used sort of the systemic priming model followed by an intranasal challenge. And in our study, we really used all intranasal as the priming. And I really think that is the fundamental key point, is that how the allergens are initially sensed. And I think this really ties in well with the work that Carrie just discussed. How the allergens are initially sensed plays a critical role in the entire process downstream. And if based on her work, you can clearly see how which dendritic cell migrates to the lymph node and what those dendritic cells are making is going to play a really important role in the downstream T cell responses that you're going to get. We haven't looked at the Th9s. Obviously, our phenotypes are very strikingly different from that work. It's something that we plan to go back and do, but I do think that the role of IL-9 in general in allergic disease is still a little bit it's still a little bit unclear, and my suspicion is that it will have a lot to do with tissue specific and context dependent responses.
Thank you. So we have a question from Gary. He asked, nice work. What is the cellular source of IL-10? Could it be an autocrine signaling?
Yeah, this is a great question and something we're actively working on. So IL-10 is very pleiotropic. It could be coming from the dendritic cells. It could be coming from the macrophages. It could be coming from the T cells. It could be coming from the B cells. So all options pretty much feel on the table. So we're in the process of both trying to identify the sources of IL-10 using an IL-10 reporter mouse. We have IL-10 floxed mice that we're almost done crossing to a variety of CREs to eliminate IL-10 signaling from different populations of cells. And then obviously our goal is to understand which of these is the key population that's supporting Blimp-1 expression. I wouldn't be terribly surprised if it ended up being more than one answer. And I think that's going to be something that's going to be a little bit tricky for us to parse out. But yeah, you raise obviously the obvious next step and something that we're really actively working on at the moment.
Thank you. I don't think there are more questions. So yeah. Thank you so much, Amanda, for this great talk.